Good day, everyone. Thank you for coming. This is a proposal presentation, so any feedback you might have will be greatly appreciated. So when we look at Bombyliidae, it's an incredibly diverse family of approximately 5,000 recognized species, and in the Afrotropics, a landmass that covers 20% of the Earth's surface, 30% of the recognized bombyliids occur, indicating there's very high recognized diversity in this region. And when we look at the pollination ecology of Bombyliidae, particularly in South Africa, while research is limited, it does show that Bombyliidae play a significant role in floral assemblages in one of our biodiversity hotspots, the Greater Cape Floristic region. In South Africa, the succulent Karoo biome and the Cape Floristic region are considered two separate biodiversity hotspots. However, the Greater Cape Floristic region, or the GCFR, encompass these two regions, and therefore I'm going to treat these in this project as a single hotspot. The second biodiversity hotspot, the Maputoland, Pondoland, and Albany region, is our second hotspot along the East Coast. And each of these hotspots have well-documented records of flora. However, research into insect diversity and endemism lags behind. In the Greater Cape Floristic region, research is still ongoing into the diversity of certain insect groups, particularly Coleoptera, Lepidoptera, and Nemostrinidae. However, insect surveys in the MPA Maputoland, Pondoland, Albany region are virtually non-existent. If there are a few surveys, they're largely broadly anthropod diversity surveys that have taken place in single conservation areas, and one investigation in particularly the Mkombati Nature Reserve found 18 new species to science out of a sample of 425. And this really indicates that there's an underestimated diversity within our hotspot on the East Coast. And so I'm proposing a widespread survey of Bombyliidae within both hotspots. So the aim of this project is to survey Bombyliidae diversity and then determine whether gamma diversity between the hotspots is significantly different. This will contribute to contemporary collections of Bombyliidae in national collections and collect fresh specimens to revise a common genus called Astrolochus. This is just a sampling methodology outline, and I'll start by sampling within each biome of our various hotspots. So 4 conservation areas per biome in each hotspot will be sampled. Each vegetation type or habitat type will be sampled in each conservation area with 5 replicates of 1 kilometer transects. Habitat types will be excluded if they're considered inhospitable to bee flies or Bombyliidae. And targeting each habitat type will really ensure that we're collecting the broad-scale diversity and not undersampling specimens, and will reduce the possibility of missed species. From there, I'll identify specimens to species level, and each morphospecies collected per conservation area will be sequenced to generate a general barcode database. And this will also help to just pair males and females in taxonomy or species descriptions where species have only been described of a single male or a single female. And from there, I propose using a multi-species occupancy model to investigate the differences between diversity in the hotspots. And this model estimates how many species, using presence data, estimates the diversity, even if some species were never detected during surveys. It accounts for imperfect detection by modeling whether each species is truly present or absent in the community, how likely each species is to be detected during field surveys. And in this project, we'll use the hotspot as a covariant, as well as including vegetation type, elevation, biome as covariates. And it'll estimate the total number of species or gamma diversity in each hotspot and identify species that are more likely in one hotspot than the other, and directly test whether overall diversity differs between the hotspots. And this approach provides a more accurate and statistically rigorous comparison than simply counting observed species and helping reveal hidden diversity patterns important for conservation planning. So what I expect from this survey is I don't think there's going to be significant difference between the hotspots in Bombyliidae diversity. This is merely based on literature reading, where we see that there is unsampled Bombyliidae diversity in the East Coast, but we're seeing that there is a current pattern, particularly when you look at open source databases like iNaturalist. And when we look within the biodiversity hotspots, I do think that we'll see a higher diversity in the fynbos within the Greater Cape Floristic region and the savanna biomes within the MPA. And this is strictly based on the current knowledge of diversity within these hotspots. And I've mentioned taxonomy. Within South African Bombyliidae taxonomy, we have what I've termed the Hess problem. Hess published a notorious revision in 1938 and 56, and this is still the current recommended taxonomic keys. Even if there's been higher level revisions, particularly at the genus level, you still have to go back to Hess's work to identify your species, and often there are sex representatives missing, or species have been described of a single specimen, which means that your intraspecific variation has not been captured. And so, considering this, I believe that the barcodes are going to pair males and females, and if you have a male that's considered one species A and a female considered species B, the barcodes will reveal that these are the same thing. And so, considering that, it's likely that the diversity of Astrolochus is going to come down, and there is a possibility of collecting new species, but I think considering the current literature, we'll have synonymy as well as description of new species. And with our outcomes, it's going to expand the current collections, as I've said. It'll be the first widespread survey of Bombyliidae to investigate diversity between the hotspots. For each protected area that I sample, I'm going to provide a checklist to the respective authorities so they can take it under consideration for conservation planning. And then any new species that are collected will be formally described, as well as the revision for Astrolochus. So thank you to all the funding bodies so far, and thank you for listening. I really appreciate it.
Thanks, guys. Any questions? Yes. Yeah.
Matt, thank you so much for your wonderful presentation. I'd definitely say go for it with this research. So, with regards to your methodology, I like how you set up like the sites for each locations, like around South Africa. But how you planning to capture the Bombyliidae? Do you plan just to do like direct observation of them, or you plan to maybe set up a pan trap or pitfall trap to capture the specimens? So like how will you do that?
Simply hand netting. They're quite difficult to catch. They apparently don't work in Malays traps. There has been some observations of them falling into pitfall traps, but it doesn't really work. So it will be walking along the transect with a hand net and collecting them as I see them. So it's going to be a lot of intense field work, but right now, from my experience, that's the best way of collecting them.
Awesome, thank you.
Thanks, Cass. So with your species occupancy model, is that estimating diversity, or is it estimating species richness?
It is estimating diversity. I haven't had a chance to actually work through the model myself with a simulated data set, so I can't really speak to the specifics as yet, but all the papers I've read, they've compared species diversity between two sites, and they use species richness for that.
Okay, but I mean, I guess if you're going off of presence-absence records, then you can't capture like whether certain species are more abundant in certain parts of the landscape than others.
That's a different part of the model. It's called a multi-species abundance model. And I can do that. It is something I've considered, particularly when we think about the mass blooming in the Greater Cape Floristic region. Off the top of my head, I think Mary Besonnet is going to be the most abundant.
Right.
But it would be really interesting to see how that would then compare in the MPA, because there's very little known about the Bombyliidae diversity and what's abundant. And what's really exciting about this work is that it's then a step into ecology. Mm-hmm. because once we know what's there, we can then study how they support the ecosystems that they occupy.
Thanks.
Any other questions? All right. Thanks, guys.
Thank you.